I'd like to just thank all of you for being here today, and special thanks to Senator Reid and your team for all the support, and to our distinguished panelists and moderator. So in the next hour or so, let's take a journey together into the fascinating and complex world of RNA. So we used to think that the RNA is just an identical copy of DNA, but we now know that it's much more complex, and it is features with as many as 200 components, as opposed to 4 in the DNA. So today, we still do not have ways to read the RNA with all the 200 components. And just to illustrate the problem, I invite you to look at our agenda for the day. And on one side is printed with letters missing and making it hard to read. And on the other side, it is complete and much clearer. And this demonstration mirrors the challenges that we face when trying to read RNA with components missing. So RNA is present in the cell of every plant, animal, and human. So we must have a way to be able to read the entire RNA in order to understand its functions. So there is a critical need for technologies that can decode RNA. And recently, the National Academies of Sciences, Engineering, and Medicine published a report titled Charting a Future for Sequencing RNA and Its Modifications, a New Era for Biology and Medicine. Our expert speakers today will outline the transformative power of mastering this RNA technology in fields such as agriculture, data storage, human health, and national security. This will address critical issues such as the ongoing threats of the pandemic, the more than 10,000 human diseases still without treatment, food insecurity faced by children in nearly 10 million households in the United States, the increasing energy demand for data storage, and maintaining our lead in innovation and securing us against national threats. So I hope that you'll find today's session informative. And with that, I'm going to turn the microphone to my colleague, Mukesh Jain from Brown University. He's the Dean of the Warren Alpert School of Medicine.
Thanks very much, Dr. Chung, and good afternoon, everyone. Thank you for being here. It's really great. As you just heard, RNA is not well understood, yet it has enormous potential, and you'll hear about that from our 4 distinguished speakers. And so I have the pleasure of introducing them as well as our moderator. And I'll start with Dr. Drew Weissman to my left. He's the professor in vaccine research at the University of Pennsylvania and co-recipient of the 2023 Nobel Prize in Medicine for discoveries that enabled the modified mRNA technology used in vaccines such as COVID. To his left is Dr. Olgika Milenkovic, professor of electrical and computer engineering at the University of Illinois, who has pioneered methods to use DNA and RNA for data storage. Moving along, to her left, Dr. Brian Gregory, professor of biology at the University of Pennsylvania, who's focused on making plants more resilient to climate change. And then Mr. Ryan Morhard to Brian's left, who is the Senior Director for Policy and Partnership at Ginkgo Bioworks, where he engages on issues at the nexus of biotechnology, sustainability, health, and security. After the 4 talks, we'll have a short panel discussion moderated by Dr. Frederick Tyson, Scientific Program Director at the National Institute of Environmental Health Sciences. But before we get started, we'll have introductory remarks from one of this nation's most distinguished public servants, Senator Jack Reed. Senator Reed is a native of Rhode Island, a graduate of the U.S. Military Academy at West Point, as well as Harvard Law School. He served as a member of the House of Representatives from 1990 to 96, and since 1996 as Rhode Island's senior senator. He has major roles in the Senate as a member of the Armed Services Committee and the Appropriations Committee. He is also a strong advocate for health, particularly child health. And he is a strong champion for the potential of RNA for health and beyond. And he has offered Dr. Chung and I incredible support and sage guidance over the past few years. Senator Reed, thank you for being here, and we look forward to your remarks.
Well, thank you very much, Dr. Jane, and I'm fortunate the microphone is not working. You see how much taller when I'm sitting down, so I just feel great. Welcome. This is a very important briefing because we are on the verge of, and borrowing the words of John Kennedy, a new frontier, and we have to be the leaders in that frontier. I want to give a shout out to Fred Tyson, too, not only for his moderation of the discussion. He's originally from Providence, Rhode Island, correct? And he probably still knows people that vote there. So, Fred, hi. I also want to thank this distinguished panel. And I must say, distinguished is an understatement. This is an extraordinary group of men and women who are going to share their insights with you. I also want to thank the Warren Alpert Foundation for supporting the RNA science by funding the National Academies of Science study that began to chart the path forward for us on RNA. Today, you'll get a crash course on some of the nation's foremost endeavors from the nation's foremost researchers in RNA. You'll be hearing about how innovation in biology, medicine, and computing related to RNA will advance healthcare, our economy, and our national security. You'll hear about how RNA research can help protect our food supply from the impacts of climate change. And you'll see how RNA can play a role in thwarting bioterrorist threats and even cyber threats. In fact, one of today's speakers will be talking about how RNA can be used for the safe storage of personal and business data. As you see, the expanse of RNA is something that offers huge potential. And as a nation, it is essential we maintain our global leadership to harness this tool for good. One of the best descriptions of new, breathtaking technology I heard was years ago when a president of a university said, "This type of technology makes good things better and bad things worse." Our job is to take the good and to avoid the bad. Thank you all for coming today. Good luck. Thank you very much.
Thank you very much. So what I wanted to talk to you about today is the potential of RNA for health. I've condensed it to 10 minutes. So if I leave out your favorite disease, I apologize. Next slide. So this is the timeline. And I put this up because I spend a lot of time talking to lay audiences who are hesitant to take RNA vaccines. And one of their comments is always, well, it was invented in 10 months and I just don't trust it. RNA was discovered in 1961. The first time RNA was put into patients was in the late 90s. It is not brand new technology. Next slide. To me, this is an incredibly striking representation of what the COVID-19 RNA vaccines did. These are predictions of the number of lives saved by the COVID-19 vaccines. People have calculated it's something around 17 million. But this shows across different economic status countries what effect COVID-19 had and how much vaccines, how many lives they've saved. To me, that's the most important thing. Next slide. So there are an incredible number of uses of RNA. They can be used for a variety of vaccines. They can be used for gene therapy. They can be used for treating acute and chronic diseases. The potential for RNA is enormous. Next slide. This is just a very brief list of the different types of vaccines in development. They're being developed for just about any infectious disease you can imagine, from malaria to tuberculosis to HIV, influenza, and many others. But it's not just infectious diseases. Vaccines are being developed for cancers. They're being developed for autoimmune diseases. Why that's critical is currently the way you treat an autoimmune disease is with an immunosuppressant drug. You poison the immune system. It helps with the disease, but it makes people susceptible to infections. Vaccines have the potential to suppress specifically the autoimmune disease. It's also being developed for allergies, things like food allergies, peanuts, and aeroallergens like dust mites. So the potential of vaccines is enormous. Next slide. For gene therapy, why RNA is so important. On the right is how gene therapy is typically done. There were two sickle cell gene therapies approved last December. You take cells out of the patient, you culture them, you infect them with a lentivirus, you give the patient chemotherapy, and then you give the cells back. That's days in an incredibly sophisticated hospital. It supposedly will cost about $3.2 million per person. 300,000 people are born with sickle cell every year, mainly in Africa and India, but across the world, 100,000 a year in the US. 3.2 million times 300,000 isn't a number that our Congress considers. So the alternative is to do the gene therapy in vivo, and I'll show you how RNA can do that. Next slide. So I excuse the science, but what this is really shown, I put up is this is in vivo gene therapy, and the red is treated, the black is untreated. You can see enormous levels of effectiveness. 70 to 100% of cells are gene corrected with in vivo gene therapy. Next slide. This is just a partial list of all the different things you can do with bone marrow stem cell gene therapy that range from fixing the thousands of genetic diseases, starting with sickle cell, thalassemias, all of the immune deficiencies, moving on to other genetic diseases. It can be used in bone marrow transplants, which are a mainstay of cancer chemotherapy, and many other treatments. Next slide. CAR T cells are the new frontier of cancer chemotherapy. They take liquid tumors, lymphomas, leukemias, that previously had incredibly low survival rates, and they typically have efficacy rates in the 60, 70, sometimes 80% range. They have changed the treatment for leukemia and lymphomas. The problem is that in order to make a CAR T cell requires 10 days of sophisticated hospital maneuvering. A single dose costs $500,000. Next slide. With RNA, we can make CAR T cells in the body. So the only thing we have to do is give people an off-the-shelf RNA LNP, and that creates CAR T cells in the body that treat the disease and go away. We don't know the cost of this yet, but it's not going to be $500,000. Next slide. Heart disease is the leading killer in the world. These are all the different manifestations. The problem with heart therapeutics nowadays is they're mainly directed not at the heart. They're directed at high blood pressure, they're directed at the kidneys to balance volumes. There aren't good heart therapeutics. We've developed ways to specifically target the heart and deliver RNAs to the heart that will someday be useful for treating heart attacks, for rebuilding cardiac tissue, for fixing cardiomyopathies, and a variety of other things. Next slide. These are the currently used approaches to treating the heart in heart disease. They all require either invasive procedures where you open the chest or where you catheterize people. Thank you very much.
The world is full of big data, that you will be able to squeeze all that data on a disk or on a flash memory, then you're wrong, because we are talking about petabytes and terabytes and zettabytes, and you really want to have more volume for your data problems. And this is, for example, how a server farm looks like. It's not something that you can take with you. It requires extreme energy consumption to maintain these server farms that store data. They're sometimes not long-term compatible because the data storage technologies change. They tend to break down. We have durability problems. There are privacy and safety issues, and you cannot do computing on these server farms at all. And the recent National Security Commission in Biotechnology briefing from 2023 outlined an idea that biotechnology can step in, and then that we can use molecular storage systems, systems that would store information in molecules such as this beautiful looking RNA here. And if you had some class in biology, elementary class in biosciences, you would know that DNA you can view as a double-stranded or double helix combination of strings, which uses 4 symbols or 4 types of beads. Why RNA is very similar, it's just one strand, and it has a bead that differs from the bead that the DNA uses. And the interesting thing is, if you would look at the mass of the DNA that you have in our, that each one of us roughly has in their body, that would be enough to store pretty much all the data we have on the planet right now. All of that could fit in the DNA or RNA of our organism. And here is an example how you would do storage in DNA and RNA. What you see here is a photo of one of my favorite and youngest, Franklin, via cat, because 9 billion photographs online are cats. And I probably contribute 40 images per day to that growing database. And how would you store the image of this good-looking feline? You would take the 4 types of beads that I mentioned, you would convert them into zeros and ones, 2 options for both coordinates, and that takes only a nanometer, 10 to the minus 9 in length. And then you would take the digital information, convert it into the symbols of the bead alphabet that I mentioned for RNA, stitch these together, and then create the image of this cat. And there are a lot of companies in this country that can do this for you, but at a hefty price. I tend to use IDT, one of the many companies that do it. And the cons of this approach is that we currently don't have really cheap methods for creating the beads that we use, and we have delays when creating the content because, as I mentioned, every bead will store 2 bits, 2 zeros and ones, and that takes roughly several seconds. That's unacceptable if you want to store large volumes of data. So how does RNA come to the rescue? RNA comes to the rescue because RNA has many more beads, and let me call them chemical modifications. You have something called methylation, you have a bunch of other changes, and the number of beads all of a sudden grows to 200, 300. And that increases information theoretically, as we would say, the density of your storage system, and it also enables you to do storage of more symbols or bits at the given amount of time. So what do we need to achieve this process of storing information, writing information in RNA and DNA? We really need to make the production process cheaper. And we need collaborations between chemists, engineers, physicists, computer scientists, and others. So once we encode the image of the cat into DNA or RNA, then we need to read it. You can't just store something without ever being able to read it. And currently, there are very few technologies that allow you to read the content. And the most promising one is what we call nanopores. And nanopores really try to read the content using devices that are the size of a flash memory. So they're portable, but they have issues because the content, to read the content, you will end up getting a lot of errors. So you need help from people that know how to correct errors. You need machine learning. That word, those two words always come up. Current companies use deep learning methods to really decipher the content of the data that you're trying to read, and you get errors. So you will end up potentially getting a smudge on the top of the cat, which you don't really want to have. And what we need in this case is really specialized reading devices for RNA and for the chemical modifications that are, as Vivian mentioned, prevalent and come in many shapes and forms. And here you would also require a lot of involvement from the machine learning community to enable extraction of the useful signal you have. And to just mention a few more things, a combination of DNA and RNA is probably the best way to go because it enables multiple functionalities that go beyond storage. For example, you can mimic something that those of us that worked in storage devices call rank modulation that is sometimes used in flash memories. And rank modulation allows you to basically combine DNA and RNA. You use DNA as a substrate on which you grow what we call RNA tails. And those RNA tails can have different lengths, different compositions. They can be random, which is good for security purposes. And you can even use it directly on patient data and just encode metadata on top of the double-stranded DNA content. You can use it for watermarking, copyright protections, ownership protection. How? Because you can encode these or add these RNA tails on double-stranded DNA at different locations with different lengths, and only the owner would know where those locations are and what the lengths are. And here you're protecting the data by making the adversary work very hard to mess with the watermark, so much so that it pays off more to pay a company to synthesize their own image rather than to tamper with the material. As a final remark, we don't have issues with uncontrolled synthetic DNA and RNA for applications like the ones that I mentioned, for the simple reason that there are randomness tests that can be used to prevent tampering with synthetic DNA and RNA. So thank you so much. And
what I want to begin with is by saying I should have actually looked at Drew's talk a little bit more closely because we could have had a really collaborative effort whereby he would have said something in humans and then I would have come right after him and said, we can do the same thing in plants. Okay, and so in 50 years from now, us nor any other country in the world is actually going to meet food production net needs with our growing populations. And much of this is because we lose billions and billions of dollars a year in crops, both to pathogens, as well as now in this era of global climate change, severe weather patterns. Plants can't move, so they can't get away from severe weather. Plants can't move, they can't get away from pathogens, they can't go get their own medicines. So we have to come up with the solutions to fix these problems for our farmers and for both at the subsistence level as well as those that are in the production level. So what you can see is interesting data that shows the billion-dollar losses across the US from 2023 and then, in 2022, and then in fact, 10 more of those events just a year later in 2023. We lost over 20 billion in crops due to just 20 weather events in 2022, and then we lost $10 billion more, $30 billion And this is only measuring the billion-dollar crop losses. This is not looking at those subsistence farmers and those farmers that work on the less than billion-dollar type of level, okay? And so what this is gonna ultimately lead to, like I said, is massive global food security issues, not just in our country, but across the globe. And in fact, it's gonna start in Africa and Asia first before it moves to the US, because our production right now is above our per capita, but it's not gonna last more than 25 years. And so then plants can do everything that humans can do, and specifically we can put RNAs on plants. They will uptake those RNAs through little cells known as stomata, which they use for gas exchange, and then they can take these RNAs and turn them into their own internal medicines to respond to viruses, bacteria, fungal pathogens. And what we're also learning is we can also program these plants just by spraying RNAs onto them to also respond to the various types of abiotic stresses, including drought, salt, and all of the other things that they see in the field. And so we can do this, like I said, just by spraying naked RNA or by putting RNA through clay or other mechanisms by putting those RNAs onto plants. And not only can we do this to make them respond to stresses and pathogens, but we're also, as Drew said, we're now trying to improve crops both at the level of production as well as the type of vitamins and minerals that they put into their food. And we can also do this, once again, by programming those plants by spraying specific RNAs onto their leaves such that we can improve the quality also of the crops that we produce in the field. And thank you to Olga for introducing you guys to RNA modifications and to say, actually, we don't even have to target the full RNAs. We can actually just target a very specific subset of RNA modifications to increase crop production, as well as make these crops more drought and abiotic stress resistant, as well as more resistant to pathogens. But in the future, what we want to do is make these RNAs at very, very large scales, targeting both abiotic stresses, pathogens, as well as improving those traits in the crops that they produce. But right now, as Olga said, making these types of RNAs is not where it needs to be. We need to vastly increase the ability to produce these by funding collaborations between scientists to improve our synthesis, as well as improving the actual industrial complex to make the RNAs at scale, such that we can democratize RNA, both for those large-scale farmers, but also for those subsistence farmers that cannot pay right now the amount of money that it takes to synthesize their RNAs to improve their crop production, as well and to improve the quality of the crops that they do produce. So we need to improve our funding for this such that we can democratize this and use this across the spectrum of the farmers, not just, well, focused here in the US, obviously, for the purposes of this panel. At our dinner last night, we were talking about South Korea. They're putting $2 billion right now into RNA synthesis for agriculture as well as health purposes. And I can tell you that's far behind what China is actually doing, because they've already moved into being the world leader of making stress-resistant and pathogen-resistant types of crops. So we're already way behind. And that technology, we need to move forward by improving our RNA technology for crop production and making these stress-resistant crops as we move into the future. This is where we're headed. This is actually where we're at, but we need much more funding, both for the basic research as well as for making the production of RNA much easier and at much larger scale, such that truly we can take a spray can, move it into the field, whether it be a subsistence farmer or a production farmer. We need to do this for the good of the health of the global food economy over the next 10 to 15 years. Thank you guys for listening to me, and I'll take questions later.
Good afternoon. Thank you. Wanted to first, of course, thank Brown University and Senator Reed's office. But first and foremost, what I hope to do is, contextualize a little bit as we wrap up the panel, contextualize this RNA revolution in the context of a broader biorevolution. And we've already heard today just a couple of applications in health, in agriculture, in data storage, and there's many more. There's many applications, and the fact is that we are sitting in a moment where changes in biotech R&D, changes in AI, emergence of other data science, et cetera, are creating brand new opportunities with actually transformational applications, we've already heard a few, across the economy. And when you have a technology that offers transformational potential, it of course has economic and national security implications. We've seen that. And so countries are competing. Okay, the US is competing. The US has prioritized this and should continue to do that. China is competing. The European Commission a couple weeks ago referred to biomanufacturing as potentially one of the most promising technologies of the century. Okay, so the world is looking at this as a potential next wave of technologies. And the question is sort of why? Well, why? One reason is biology is not only offering brand new ways to treat and cure diseases that affect all of us, but it's offering, as we just discussed, it's offering a brand new way to make things. And when anything offers a brand new way to make things, it actually starts to shape the sort of geopolitical underpinnings, the sort of interdependencies that affect all of our geopolitics. It starts to change all of those. And so it starts to become very interesting. Where things are made and how they're made really matters. And experts have looked at biotech, and they've looked at these trends, and they said, wait a second, it looks like you could use biotech to make more than 60% of the physical inputs into the global economy. That's a big deal, especially when most of the things that we make so far are coming from raw material extraction or from biodiversity or coming from fossil fuels, okay? So biotechnology offers a chance to change that, and it's becoming very interesting also to companies. Companies who, especially coming out of the pandemic, are saying, wait a second, how can we make our supply chain more secure? How can we also thrive in a multipolar world, right? Companies are looking at that. And also, how can we start to reduce emissions or become more resilient against climate change? Biotechnology and biomanufacturing offers a path forward there with the right tools, right? So that's why we're discussing investing in RNA technology, right? So, but the question, I think, if I was sitting where you're sitting, I was saying, wait a second, I took biology in high school and none of this was discussed, right? Which is true. So why is that happening now? I think it's worth zooming out a little bit. Why now are we saying, hey guys, bio is the solution? No one said that 15 years ago, really. Maybe some really smart folks in this room, but basically we weren't talking about biology that way. We've talked for a long time about biology and healthcare. That's the first thing we all think about. Well, why is that? Well, basically, it comes down to tools. We're discussing here tools. The tools of biology basically haven't changed in decades. It's surely, you can imagine, it's a scientist sitting at a lab bench, moving liquids around and waiting and seeing how the results go and jotting them down and sharing them with nobody. And then someone else tries again. It's completely commercially impractical for most applications outside of pharma, historically. It's expensive and commercially uncertain. You're going to buy a lab, hire a bunch of people, do a bunch of experiments. They might work, they might not work. That will work pretty well in pharma. That won't work for a lot of ag stuff. It won't work for data storage. And it won't work for materials and chemicals and personal care and food, et cetera. But that's changing. Now what we're seeing is 4 trends converging. We're seeing automation. We're seeing tools of reading and writing and editing DNA, right? That's things like DNA sequencing we just referred to, but CRISPR, which you may be familiar with from the Nobel Prize, right? Reading and writing and editing DNA. And then the tools to do that are becoming cheaper such that you can generate data, lots of data, such that you can start to have data-driven insights, right? Which feed into also the final converging tool, which is AI. And altogether what these tools are doing is they're coming together to make it so biology becomes more programmable. Basically what that means is we're not doing an R&D experiment that's maybe works, maybe won't, maybe takes for a long time. We're starting to use biology to do exactly what we want it to do. And when that happens, it becomes a lot more interesting. And also, we're seeing the emergence of platforms to do that so people can start to use biology without having their own lab, without having a PhD, and without taking 12 years to do that. They can start to sit at their internet browser, type in what they need, type in the sequence that they think they want, and all of a sudden they have the application coming in the mail. That's a vision of the future, okay? But let's just emphasize a little bit, because I'm here kind of speaking to the national security implications. To do that, to actually unlock that world where you're sort of seeing an AI-enabled biotech revolution driven by big data and high-level automation, things like this, you start to see the relevance of these kinds of interdependencies that we've seen in other areas of technology. For example, all of our semiconductors we've seen, these are coming from Taiwan and Taiwan Semiconductor, right? So that you're going to start to see in biotech, in that world, where the hardware is made really matters. Where that large facility doing the large-scale biological data generation and the large-scale experimentation to feed the AI models, where does that exist? Who's designing it? That's going to start to really matter. For example, I work at a company called Ginkgo Bioworks. We're sitting in Boston. We have 300,000 square foot facility and another 200,000 square foot plan to come online soon that's just doing basically a biological data factory, right? When we're not aware of another facility of this kind outside of China. So these are the kinds of fixtures that will exist in a future bioeconomy that unlock applications in the same way that chip fabs also enable a digital economy. And it's worth thinking through those things because those will be the crucibles for not only unlocking applications, but for developing the hardware and the tools. And so I highlight that to say that there's trends and they're making biotechnology a technology of national security. The final point is before I started speaking, not only were you biology in high school, but also it's I thought bio and national security were things like COVID and biodefense. That's true too. So it's also worth thinking about that tool. As tools begin to advance, you will see increased risk as well. It's really important that in a bio-revolution that we have biosecurity in the same way that the digital revolution had cybersecurity, okay? In part because if you're really good at working with biology, we've just seen with mRNA, you can mitigate risk and impact of biological threats like pandemics. But also, if you're really good, you can control the norms and values and standards behind and to safeguard a bioeconomy that we have really benefited from in the digital economy. So leadership absolutely will matter, and I really appreciate this folks in this room and their ability to affect that leadership, but also the team at Brown and the National Academies for the chance to speak to this issue, because this is really first and foremost at the front lines is RNA as we move forward. So thank you so much.
I'd like to take this opportunity to once again thank all of these speakers for these really powerful and insightful presentations. I learned a lot more than I thought I would even in this short time. So I'd like to ask each of them a series of questions, and I'll start out with Drew, and I'll ask all of the speakers these questions. So Drew, starting with you, what is the most exciting thing that the development of RNA can bring to your field?
So I don't want to pick a particular disease, but its expansiveness and its therapeutic potential is enormous. So it's not going to be all of those diseases, but a significant number can be targeted and will someday have RNA-based therapies for them.
Thank you. Ogega, I'll ask you the same question. What is the most exciting impact RNA can have in your field?
So my field is really information theory or data storage and correcting errors in communication and storage systems. And the fascinating thing about DNA and RNA is that you realize that you can implement a lot of the paradigms that we came up completely independently through a different thought process using these macromolecules. I will give you as an example that very recently people have realized you can do in-memory computing, where you don't have to download the data, process it, and then store it again, which is very time-consuming and expensive, obviously. You can do it inside the memory as well, directly. That has been achieved, let's say to a limited extent, but it's possible to do it through the use of macromolecules such as DNA and RNA. Then you can ensure, you can come up with a large number of data hiding protocols. Just imagine you take a piece of RNA, you encode the information that you want to be a secret, and then you can put it under your fingernail, you can put it on a stamp and send it out, and no one would know because RNA is everywhere. So it's a fascinating, if you just sit down and think of everything we achieved nowadays with communication and storage technologies, there is always something that you can do better with macromolecules such as RNA and DNA.
Okay, thank you for that response. Brian, I'll ask you the same question.
Yeah, so I think I made this point, I hope, abundantly clear, but obviously our most our biggest goal here is to make climate-resistant crops, right? That's our number one. That's what's been driving my research since I started at Penn, and since I started working on plants. But to take it back more from a societal perspective when it comes to agricultural production, the ability to use RNA basically as a plant medicine really democratizes the agriculture, we think, right? And so that's the perspective that I like to look at it is if we can get RNA online really to do everything we think we can use it to, we can democratize its uses both for those subsistence farmers as well as production farmers. And that's ultimately what I want to get to, right? Is not big ag driving big production farms, but now those farmers that are not big production farms, but low profit or subsistence, let's make this technology also available to them. And I think that RNA has the ability to get climate-resistant crops across the board to all farmers.
Okay, thank you. And Ryan, the same question to you.
Yeah, it's hard to think of anything more exciting than feeding the world and curing all disease. So I think these are all very exciting. I think but if I had to answer beyond that, I think there's kind of two things that are really, I think, exciting. One is what we don't know, right? And so I think you think a little bit about when the Human Genome Project was started, really created conditions that sort of facilitated the whole bio-revolution and U.S. leadership in that space, and all sorts of applications that probably, if you went there, and some of you may have been there at the time, maybe didn't anticipate completely. And so I think that part is exciting if we would just allow that. And I think we've also seen in that space the benefit of leadership in brand new categories. And so I think what's exciting there is this sort of the unknown and the chance to kind of solve big problems. I think the other exciting thing is that I suspect that there's lots of people in this room who, aside from getting a free lunch, did not think that they would be spending a lot of time just thinking about RNA. And I think that is what we need to sort of create a condition for, which is all of us, I think, feel very comfortable talking about digital tech. We use it every day. We don't need a PhD to talk about digital tech. But I think many of us, at least many people in the world, feel like they can't engage on bio because they don't have a PhD. And that, if we keep having these conversations that are application-based, where people really care about that stuff, and we have tools for it, they're not going to really care about whether they have a PhD or not. They're just going to solve the problem. And I think that part gets really, really exciting when you think about what that could mean for actually, changing the conditions that we're living in.
Okay, thank you. We'll go in a slightly different order, so I'll ask you, Olgiga, first. What do you need most in your domain to make significant progress with RNA?
There is a politically incorrect answer, which is money. And then there is a politically correct answer, building blocks for RNA technologies that will allow us to read RNA. Then RNA methods in artificial intelligence and machine learning that help us make sense of RNA data that is collected from living organisms. So there is a long wish list of what we would like to know about RNA, but without the technology, biotechnology, to be able to create RNA content, be it for medical applications or plant sciences or for storage and computing, and then being able also to read that content in an accurate manner. Accuracy is a big issue. Every single modification counts, and if you miss a few of them, you can come to very wrong conclusions. So accuracy and quality of the readouts is very important. So personally, I would love to have let's say, Oxford Nanotechnology nanopore that would give me almost perfect 99.99% accurate readouts of RNA data, so that I don't have to use error correction coding and add overhead, which costs even more. And obviously, I would like to make the technology cheaper, because the technology is there and it's not science fiction. Sometimes it sounds like science fiction when you talk about storing information in RNA and DNA. The technology is there, it just, we have to pay a large amount of money, which is not a good idea when you're in academia. You run out of funding pretty quickly. If that can be made less costly and more readily available, that would be a great, push for biotechnology.
Well, it sounds like that the politically correct and politically incorrect answers are, yeah, they go hand in hand. They're weighed together, and you can't get away from one without the other. So, Ryan, I'll ask you the same question. What would you need most in your field to make significant progress?
Well, I'll build on Olgika's points because if you had an effort to go out and generate RNA data and you really had a big ambitious initiative, it would be a sort of, it would pull the development of those tools, right? And also improve efficiencies and reduce costs. And I would suggest that one way to sort of think about that initiative is when we look at the big AI tools that we're all probably using, ChatGPT and the others, those were trained on basically internet and social media and Library of Congress data. Do you need the data? And there's not a Library of Congress for DNA or RNA data. There should be. I suspect there are some countries working to develop those types of databases, and those countries will lead. And so the US should do that, and it could focus on DNA and RNA data, and in doing so would be a real pull to develop the tools of both for DNA and RNA sequencing and storage, and would really, I think, create conditions for lots of innovation.
Thank you for that. Drew, I'll ask you the same question. What do we need most to make significant progress in your field?
So I apologize, but the top three are money, money, and money. If you go beyond that, as an academic scientist, my hope is that the United States in general in the education system ranks science as a more important entity. The days of American research scientists, research academics is going down. I don't know how to reverse that, but I think we need to realize the importance of science in general, leading to technology, leading to development, leading to the betterment of the world. And we need to prize that and increase that capacity in the US system.
Okay, thank you. And Ryan, the same question. I see if we get a different answer on this one.
Yeah, so I mean, to summarize Ryan and Olga's points, I mean, obviously we need money, and that's for basic research, right? We need to start with basic research. The one thing I didn't hear, because I really do want to echo Drew's education points, mainly because I have two young boys being educated in our system, and the science education is dismal in this country. And STEM nights aren't going to cut it, right? One STEM night a year is not going to cut it. When I go and try to talk to the public about agriculture, they get the idea of like stress-resistant crops. But when I then use the word RNA, the acronym RNA, the eyes glaze over, and there's basically no basic understanding of one of the fundamental building blocks of everything biological. So that's a massive problem. The second thing I didn't hear here that I wanted to really focus on was the manufacturing side, right? We need better infrastructure, and we need more focus on these manufacturing type of hubs, not just Ginkgo, but more type of hubs, right? And maybe not also just private companies making these hubs, but a much, much more democratized type of hub, right? That is government funded, such that we can synthesize the nucleotides, the chains of RNAs to make that spray can of RNA a reality, right, for the field. Like, we have to have the infrastructure to make that type of product, to get it out into the field for all applications that we're saying here. Because as you see, once again, private company IDT synthesizes our chains to do our research, right? We don't have the fundamental infrastructure for manufacturing of the tools that we need to make all of these dreams a true reality either.
Well, I'll stay with you, Brian, because I want to ask another thing, but you may have already answered that. And that is, what is the major impediment to making progress? But I think you may have already addressed that.
Yeah. The lack of knowledge in the public, number one, right? And yes, definitely the lack of technology, both from basic perspectives, the sequencing technology that Olga could spend a lot of time talking about, and then from a larger scale, like I said, the synthesis of these things to get to the next level of taking those basic findings and turning them into products and getting them into the fields and into all farmers' hands. We need that. We need better manufacturing of all things RNA.
Okay, and Ryan, I'll ask you to follow up on that same question. What are the major barriers, you think, that are impeding progress?
Yeah, I think awareness raising and bioliteracy they're going to be really important. I think the other thing that that serves us well is sometimes being a little bit empathetic with the fact that a lot of the stuff we're talking about just wasn't technically or economically practical just like not that many years ago. And so sometimes I think we can kind of approach folks we're in a good position. We can approach people and say, hey, good news. You know that hard problem we've been struggling with for all of humanity? We think we have a good solve if we just do X, Y, and Z. Like, that's pretty interesting, and that's a good position to be in. So I think that's something we can lean into, but I think this kind of base level bioliteracy and awareness raising is something we really can't take for granted either.
Okay, thank you. And Ogiga, I'll ask you the same thing. What are the major barriers to making progress?
I wish I mentioned education, but Drew thought of it first. So I think for us that work in academia, it's really paramount that our students are creative and that they take off with the great background knowledge and start their own careers afterwards. And one thing, for example, when it comes to DNA-based data storage and computing is that we really need interdisciplinary education as well. It's very hard to recruit students. It was very hard to recruit students who would want to work in this area because they would have to know so many different areas and disciplines. And it created a bit of an issue before the whole field took off because it was hard to have a educated dialogue or discussion if you have someone who has a background in artificial intelligence, someone comes from the bio field, someone else like me worked in error correction coding and data storage technologies. So we need to obviously raise our level of interdisciplinary education. And that is the educational component, and technologically, obviously, we need to be able to reduce the cost of everything we do. It's one thing to have money. I kept mentioning money, 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 but at some point you run out of money if you don't make the technology more accessible and cheaper. And I think with the nanopore sequencing, the cost of sequencing went really down. But the bottleneck, in my opinion, is really synthesis, and everybody in the field would acknowledge that. And that is where I personally would like to see the largest investment being made.
Okay, thank you. And Drew, I ask you the same question. What do you think the major impediments or barriers are to making progress?
So I'll say money a different way, and that's support. We need, starting with government, NIH, NSF, DOD, other support that recognizes the importance that RNA has across many different fields. Once that's achieved, biotech will see that. They'll recognize the importance. They'll start kicking in their support, followed by pharmaceuticals, followed by charitable organizations. But we need a system of support that can help build this technology and expand it and make it more useful, durable, and applicable to everything that RNA can do.
Okay, thank you. I have a couple more questions that I'd like to ask, and I'll ask them actually of individuals. And Brian, I'd like to ask you, as I work at NIEHS and we're worried about toxicities of everything, what do you have any thoughts about or data about the possible toxic effects of many of the types of things that you might be treating agriculturally?
Yeah, so we've done a lot of work on this, and I've seen a lot of data because we've thought about this, right? And this is obviously what the EPA was very interested in before any of these types of things were used. There is not any evidence that they are toxic, right? Because yes, we can stabilize RNAs kind of, but inherently RNA is not super stable, right? It can attack itself, and that's why it's not our genetic material, is because it's not stable enough to be our genetic material, and that's the cell uses it for that, right? It can be an unstable intermediate, and that's exactly the way you want it, because sometimes you want a lot of protein, sometimes you don't want a lot of protein, and by making that step in the middle inherently unstable, You can make a lot of it, or you can get rid of a lot of it very, very quickly, okay? And same thing happens when you start spraying it around the field. It sticks around, it gets into the plant, and once internalized, the plant responds, but the stuff doesn't stick around in the environment for more than a few minutes, right? So the plant internalizes it, makes its response. The stuff that's around doesn't really affect anything. Now, one interesting anecdote, sorry, you set me up to tell this story, but obviously working on plants, I know the amount of RNA really in every organ of a plant, right? And this is across the spectrum. And so we as humans, because we eat fruits and nuts, which are the reproductive organs, right? The progeny basically of plants. This is the organs of plants that have the largest quantities of their own mRNAs, small RNAs, tRNAs, ribosomal RNAs. And so throughout time, right, from early hominids to now, because this is the type of tissue that the plant wants us to eat, we have actually literally been eating the highest concentrations of RNAs possible from our basic kind of food groups of those plant species. So you don't have to worry, you've been eating these things since prehistoric times, so just keep on eating them.
Okay, thank you. And actually, you touched on a point that is a good segue for the next question I had, which was about stability. And as I think about RNAs and what I learned years ago, they're not a very stable molecule. So, Ogeka, I'm wondering how that actually works with data storage when you're talking about a molecule that's so unstable.
Oh, that's actually, that's actually a good question. And I think it's good to be unstable sometimes because if you're the master store like DNA, then you're not capable of doing a lot of other things. But if you are RNA, then you are designed to be multiple things at the same time. I mean, I guess every one of us knows that there was always the question, which came first, RNA or DNA? And there is this RNA-centric view of the evolution, which says RNA came first because it could store information and it could do many other things. It folds, it can help with conversion of the genetic material into protein structures. In our case, when it comes to DNA-based data storage or RNA or in DNA-based data storage, there is really not to go into details, the sugar is what's problematic, but you can combine actually nucleotides, DNA and RNA nucleotides, and use deoxyribose sugar and get something that is much more stable. So that's not an issue at all. The other thing that I didn't manage to say when I was going over my slides was when you have so many chemical modifications and you, let's say, do something that we call enzymatic synthesis, you get a random string. And randomness is good because there were concerns similar to the question asked of Brian. What do we do about potential hazards? If a string is random, then it can pass a randomness test. And you know that that whoever is ordering that or synthesizing that RNA is not going to use it for potentially dangerous applications. So you have a lot of things that you can do with RNA, especially in combination with DNA, that makes it an extra ingredient and makes the system more diverse. On my last slide, you would have seen something related to computing, which is called strand displacement, and that's another place where RNA would have been really useful, and I can take that offline if someone is interested in hearing more about it.
Okay, thank you. I'm going to ask Vivian a question. Those of you who don't know, Vivian's been the real driver behind most of this and a real force of nature. So, Vivian, I think as we're looking forward, as you look ahead, what do you think are really necessary steps for us to realize the full potential of RNA?
I'm not sure I'm the force of nature, but I am pretty stubborn. I think we all heard, right, a very difficult problem. We need to be able to make and sequence RNA quickly, cheaply. But we have people in this room, right? I'm going to ask all of you to think, before you walk out of those doors, what each one of you can do to solve this problem. We do have a telescope 1,000 miles from Earth, right, beaming back data. It seems that we should and must be able to read the RNA that is in every single one of our cells, and we have already seen the future of what it can be. Right? We all agree that we need to cure all diseases, we need to feed everyone, we need to be able to store the pictures of our cat, dogs, and beyond. Right? We need to do these things. We have public, we have private organization in this room, right? We need to work together because it will never be done. We can always complain and say, well, the government has not done X, Y, and Z, right? But we do have to do that ourselves. So anyone who is here, please contact us, right? Let's work together. And we have students in this room right now. We have students from Howard. We have students from State University of New York, right? They are our futures. And they are not going to be able to execute what these difficult problems unless we enable them. So I think my question and call to action of all of you is do something, please. You know, you've seen the future, so if it doesn't happen, we can come back and say, we could have done this, but we failed. Let's not do that.
Okay, thank you for that response. And before I turn the floor back over to Dr. Jain, I'd like to make one comment, an observation that I've made. And I think the U.S. has certainly, over the years, been a global leader in technological innovation and science. And given the broad utility of RNA, both for health, food security, data storage, national security, and so many other things, it's certainly my hope that as a nation we can forge the type of relationships and partnerships that will allow us to really maintain this type of global leadership with innovative technologies as it pertains to RNomics. So with that, I'll turn the floor back over to Dr. Jain.
Yeah, thanks, Fred. And I don't have much to add. I know we're almost at time. I just wanted to thank everyone for being here today. Your presence is really important. I do want to ask for a round of applause for our speakers who were not only just brilliant, we'll do the applause. But you saw the brilliance not only in their discussion, their science, but their ability to adapt to AV problems remarkably. We learned about the enormous promise of RNA across many, many domains of life. You heard about some, there are others. And so I hope, just to echo what Vivian said, that you walk away with a sense of urgency. Whether you come from government, academia, industry, whatever walk of life, it's really important to coalesce in an urgent fashion around something that is really, really important and maintain what the U.S. has done for many, many years, maintain its leadership in research and innovation, just echoing, again, Fred's comments. So thank you, everyone, for being here. If you have time and you want to come up and talk to our distinguished speakers, please feel free to do so. Again, thank you, and have a great day.